Krijnse, qui, comme vous le voyez sur le titre de sa présentation, travaille dans la suite finalement des travaux qui sont faits ici dans le laboratoire par Didier Raoult sur les virus géants. Et donc on la remercie d'avoir accepté de venir ici, d'avoir fait un voyage assez long finalement pour cette présentation. So we thank you a lot for the presentation. I know you understand. Okay, donc Jacomine comprend le français mais donnera sa présentation en anglais, ce qui est très bien. Merci beaucoup.
Okay, thank you for this introduction and for inviting me. Is this thing working? Hello? I don't think it's really working. Okay. Well, perhaps I don't need this. Can you hear me like this?
Yeah.
Okay.
We don't need microphone usually.
So, I realize that many of you are probably not very familiar with what I'm going to talk about today, so I will go slow and step by step and just interrupt me. And as a way of introduction, I'd like to tell you a little bit about myself first. I started really my career as a postdoc at the EMBL, which is a big European institute in Heidelberg. And I started immediately with the assembly of pox viruses using electron microscopy and cell biology. So I'm really somebody who studies viruses, cells, as well as electron microscopy. And when I started in '94, I took over the project from Beate Sodek, who looks a little bit older right now. And her supervisor was Gareth Griffiths, who was also my supervisor in the beginning, until I took over the project myself. So in 2007, they kicked me out of the institute because I'd been there too long. And then I moved to the University of Heidelberg, which is simply going down the hill. And there, since 2007, I'm heading a unit dealing with electron microscopy, and I'm also a group leader in virology, the Department of Infectious Diseases. And there's two big professors working there, one working mostly on hepatitis C virus, and one mostly working on HIV. But, so basically what I'm interested in is studying membranes. I like membranes a lot. I like, of course, virus-infected cells because they basically modify membranes so you can look at them better. And I like electron microscopy. And since many, many years, I've been studying Vaccinia Virus, which is a member of the Poxviridae. And already in the background, you can see what I'm interested in, electron microscopy and electron tomography, two techniques that I will introduce to you during my talk. And Vaccinia virus is basically a large DNA virus encoding for many proteins. And this intriguingly allows this virus to replicate its DNA in the cytoplasm. All other DNA viruses, with a few exceptions that I will talk about today as well, replicate their DNA in the nucleus. So it's a large virus. It's so large that you can actually see it by light microscopy. But we prefer to use more high-resolution techniques. And what is also very typical, and I'll come back to this also, this virus is sort of rig-shaped. Very strange morphology in a way. So, this is in short something about the life cycle. The virus comes in two infectious forms, forget about these abbreviations here, but what happens, it first has to enter the cell. We think that this happens at the cell surface. And it then delivers this core, this proteinase structure into the cytoplasm. And this core not only contains the viral DNA, but also enzymes that are packaged during assembly and which can carry out the process of early transcription. Now in this process, which is apparently activated in the cytoplasm, about 100 mRNAs are transcribed from the genome inside the core, and these mRNAs are then extruded into the cytoplasm, at least that's the idea, for translation, using the host translation machinery. So these early proteins are essential to set up this cytoplasmic process of DNA replication, which occurs in distinct sites that can be seen by electron microscopy and also by light microscopy. These sites grow, and as they grow, they move towards the nucleus. And from these large DNA replication sites, then assembly of the membrane of the virus starts. First with the appearance of these half-moon-shaped membranes, which then become spheres. And after taking up the viral DNA, basically the virus matures into this strange brick-shaped form. And this virus is infectious, but it remains inside the cell, basically hiding for the immune system. But the virus has acquired a second way of, again, envelopment by membranes, and this second membranous form is able to actively leave the cell. So amongst this complex life cycle, what I will talk about today is mostly the assembly of these membranes, and I will present you first some data that have been published, and then basically to introduce the techniques to you that we are using, and then later on I will talk about some preliminary data and also about some questions that I have that perhaps some of you may have an answer to. So, these are these crescents by conventional electron microscopy. So they are half-moon shaped, like the crescent of the moon, and we want to know how they are made and where they are derived from. And basically, based on these old images from 1968, most of you were probably not born then, it was proposed that these membranes are made de novo in the cytoplasm with open ends, so the end open. And because for us this model didn't make very much sense, we proposed basically that no, it looks like one membrane, but there's actually two membranes and you cannot see them very well by electron microscopy. And basically the membrane is derived from the endoplasmic reticulum and is a cisterna. Now why were we proposing this? For the very simple reason that this model is much more consistent with cell biological dogmas. So if you have a cell here, taken from the internet, stolen from an image, if you look at all the membrane compartments within the cell, you see that they're actually all closed. The membranes are closed. And these closed compartments are basically maintained via fission and fusion reactions with closed transport vesicles. Basically, vesicles are pinching off from one acceptor membrane and then fuses from a donor compartment and then fuses an acceptor compartment. So you can see if in cells all membranes are closed, then this model is really much more consistent with cell biological principles. So, but in electron microscopy there's two major problems. First major problem is that in order to obtain an image like this, you have to chemically fix your cells with glutaraldehyde or paraformaldehyde. So chemically fixing, then they are contrasted with heavy metals such as uranyl acetate and osmium tetroxide. Then in order to get the cells into a resin, they need to be dehydrated, which is totally unphysiological because cells contain 80% water. And then they are embedded in a resin, and in order to polymerize this resin you need to incubate, cook it basically at 65 degrees overnight. So a major question that one has to address after you obtain an image like this, is this real or is this just simply an artifact because of the embedding? Another major problem in transmission electron microscopy is that in order for the electrons to go through your sample, the sample needs to be thin. So there's a theoretical thickness of about 500 nanometers. We actually routinely work with sections that are no thicker than 70 nanometers. But if you look at an average cell and you take the diameter here across, then basically the diameter of an average cell is about 10 micrometers. So that's much too thick to observe directly. And consequently, what has to be done, as exemplified here by this orange, you need to cut slices of the cell. And then you're looking at slices of the orange. And the problem, of course, is that you lose one dimension. So you're looking at two-dimensional images of something which has a complex structure in 3 dimensions. So I have to introduce to you here two people. First of all, a very brilliant student who joined my lab about 4 years ago. Meanwhile, he went to NIH in Bethesda. He came from Czech Republic, and I hope that he's actually looking at a bottle of French red wine that got him some inspirations. And basically he said, well, we need to use more native ways of looking at cells, and we need to go in 3 dimensions. And some of the techniques that I'm going to introduce to you today are from this man here, Jacques Dubochet, and despite the fact that his name is French, he comes from Switzerland. So we need better sample preparation, and for this we use a method called cryopreservation, and we need to go into 3 dimensions using electron tomography. I'll explain this first, and then I'll come to the tomography. So if you want to freeze a biological sample, you're faced with one major problem, and that is that your biological sample has a lot of water. And if you would just dip it in liquid nitrogen, you would create ice crystals, which would severely damage your sample. So in order to overcome that, what we do is we have this enormous machine, it's about this big, and basically it's devoted to create very high pressure at very low temperatures, such that you can freeze your samples very, very quickly. So quick that you don't make ice crystals, but you make instead amorphous ice that doesn't damage the structure. So basically you high pressure freeze your sample here in this little piece here. And after high pressure freezing, after you cryopreserve it, you have 2 options. You can either use this machine here, which will at low temperatures substitute your sample for a resin. And this is considered to be a big improvement over the classical embedding. Alternatively, you can use a much more native way. Basically, you high pressure freeze your samples, they are frozen. You cut them at liquid nitrogen temperature, frozen, minus 120, minus 150 degrees. The sections here of the cells are seen here, they are frozen because we have this cryomicrotome that works at low temperature. And basically the sections are put onto a grid and directly observed in an electron microscope that allows you to visualize your sample at low temperature, at liquid nitrogen temperature. So the only thing that you've done to the sample here is to freeze it. You don't use any chemical fixation, no staining, no dehydration. Your sample is only in amorphous ice, so there's no removal from water. And consequently, we believe that this is the technique that is closest to the native state of the sample. So this is what a cell looks like after high pressure freezing and just cutting it. So you can see these amazing details in the cells. This is the nucleus, these are mitochondria, these are some endosomal structures, the ER, lipid droplets, etc. So we asked ourselves first, basically this was Peter Flanda's work, what does this crescent membrane actually look when you use better cryopreservation techniques? And most of what we did was by substitution, although I'm also going to show you some data using this pure method. And this is one of the first results that we obtained. And what you can see here is that after chemical fixation, so the classical method, you see sort of 2 amorphous lines, which become better resolved when you use high pressure freezing and freeze substitution. But even better, these layers are even better resolved when you use the pure technique, where now you see clearly 2 layers which seem to be separated by some electron-lucent space. So initially we were very excited about this because we were thinking now we proved that there is actually 2 membranes. But then when we started analyzing these layers in very much detail, we realized that only the inner layer really has the features that are typical of a membrane because of its diameter, 6 nanometers, and because it's continuous, whereas the outer layer, you can see, is sort of discontinuous and is too thick to be a membrane. And then we realized from other experiments that I'm not going to show you in detail, is that actually the outer layer is the scaffold protein of the virus. So this is a protein that assembles on the membrane and makes the membrane bend in a certain direction. Very typical for viruses, but also in cells in a way. So then we asked ourselves the question, well, could it perhaps be that this scaffold which assembles on this membrane is disguising a second membrane? So we have a situation like this where you have a cisterna, so there is 2 membranes, and because the scaffold assembles on the outer membrane, you cannot see it. And in order to address that possibility,